Hi, everyone. My name is Marc Steben. I'm the president of IUSTI Canada, and I'm very happy to be your moderator for the day. So we have a very good webinar that is prepared. So it's about a new model for gonorrhea control in Canada, both surveillance, new way of selecting best therapeutic, and maybe a vaccine. So without further ado, we'll start. And our first speaker is Irene Martin. Irene is a member of the executive. Irene is the head of Streptococcus and STI unit at the National Microbiology Lab at the Public Health Agency of Canada. So she does a lot of research on gonorrhea, and she'll give us a sense of what's happening in Canada with gonorrhea. Irene, please.
Good morning. Thanks, Marc. Just gonna, okay. Can you see my screen? Yes?
Yes, yeah, we're good.
Okay, good. All right, so I'm calling in today from the National Microbiology Lab. I'm based in Winnipeg, Manitoba, also part of the Public Health Agency of Canada. Today I'm going to give you a quick snapshot of Neisseria gonorrhea AMR surveillance work that our lab is doing. So first is a bit of background. We're all aware that the number of reported cases of gonorrhea in Canada has been increasing over the last number of years. In fact, the reported cases have more than doubled between 2014 and 2019. And in 2019, we had over 35,000 cases reported in Canada. So our lab has been doing antimicrobial susceptibility testing of gonorrhea as part of the national gonorrhea surveillance program since the mid-1980s. We determine MICs using the CLSI-approved agar dilution method. This is the gold standard method of AMR surveillance, and this image here is a plate, an example of an agar dilution plate. Every year, we publish an annual report where we report the latest AMR trending data. And this is a link here to our latest annual report. For the latest AMR trends in Neisseria gonorrhea susceptibility, here's a figure representing that. And we see that over the last number of years, the cephalosporins, which is cefixime in the light blue and ceftriaxone in the darker blue, those rates have been declining over the last number of years and hovering at about 1% in 2019. But at the same time, we see this alarming increase in azithromycin resistance. And in 2019, it was around 10%. And so that rate now exceeds the WHO recommended rate cutoff of 5%. So when an antimicrobial has resistance of greater than 5%, it's time to rethink the treatment option. Now what's interesting here for Canada, we have a lot of different regions in Canada. So the 10% is an overall average rate, but we have much more differences in different jurisdictions in Canada. And of course, all of our AMR data is used to determine the STI treatment guidelines. We share the data with the Canadian STI treatment guideline group, and they ensure that the recommended treatments are always up to date. So on this next slide, one of the biggest challenges that we have as a surveillance laboratory is that fewer cultures are available for testing. And that is because we see an increase in the use of nucleic acid amplification testing samples, which do have their pros. They are less invasive sample type, and they are more sensitive test for diagnostics or diagnosing gonorrhea. But at the same time, culture is the gold standard to determine AMR. So what we've been doing is we've been using SNP assay, which is a single nucleic polymorphism assay, which can detect mutations in the genes associated with resistance. And so with that assay, we are able to do AMR prediction with cephalosporins, azithromycin, and ciprofloxacin. Now, we're using these assays in remote regions in Canada where they can't culture gonorrhea or they can't ship gonorrhea because it is a fastidious organism and difficult to grow. And with these assays now, we're able to do some AMR surveillance in these remote regions. Something else that we're doing in our lab is using whole genome sequencing with our gonorrhea samples. And so this isn't done routinely, but we do select certain projects or certain strains of interest. And with that, we can do MIC prediction. We can determine strain relatedness as shown in this dendrogram here. We can do outbreak investigations, molecular typing, and watch for emerging resistances. So all of these efforts combined support our surveillance work and help us rapidly identify changes in AMR, which helps our work towards preventing the spread of drug-resistant gonorrhea and ensures appropriate treatments. That's it for me, so we'll move on to the next speaker.
Thank you, Irene, for this picture of what's happening in Canada. The second presentation will be made by Joanne Dillon. Joanne was previous president of IUSTI Canada, and she's moving as president to IUSTI World in the near future. Joanne is a real international expert about gonorrhea resistance, and she'll be talking to us about new ways of prescribing antibiotic or resistance-guided therapy for Neisseria gonorrhea. Please, Joanne.
Good morning, everyone. It's a pleasure to be speaking to you. I'm assuming everybody can see the screen. I have no financial disclosures. So one of the big problems that had started in the 1930s with the development of antimicrobials is the relentless development of resistance of Neisseria gonorrhea to every class of antimicrobial introduced for therapy. And that continues to this day, where sporadic isolations of isolates that are resistant to ceftriaxone or ceftriaxone plus azithromycin are being reported. The current therapies as a result of this resistance are extremely restricted. So basically it entails ceftriaxone monotherapy, ceftriaxone plus azithromycin. But interestingly, in some jurisdictions of the world, including Canada, providing that the ciprofloxacin antimicrobial resistance status is known, then ciprofloxacin could be used, providing there isn't a high level of resistance. A troubling trend, however, is the loss of capacity for phenotypic AMR analysis, as Irene Martin just described to you, across the globe. And that is driving molecular AMR test development. The loss of capacity has been due to syndromic management in resource poor environments, or the use of nucleic acid amplification tests for the detection of Neisseria gonorrhea. And in Canada, these are data from Irene's group, 85% of gonorrhea cases are actually diagnosed by NAT testing, which means that susceptibility testing is not possible on these isolates at the present time. So molecular diagnostics are really needed for the effective use of antibiotics, antimicrobial stewardship, the development of resistance-guided, i.e. individualized treatment approaches. And for that, we need molecular tests, testing NAT specimens for AMR or resistance or susceptibility, for surveillance, in those cases where we do have 85% of the isolates not tested, and even to evaluate resistance in new antimicrobial agents. The big problem with developing molecular tests for resistance is that there are multiple mechanisms of resistance depending on the antibiotic. Neisseria gonorrhea resistance is mostly mediated by chromosomal mutations in multiple genes. And depending on the antimicrobial resistance, the mutations in these genes tend to be cumulative, in other words, increasing resistance. And there are only two plasmid-mediated mechanisms of resistance to penicillin and to tetracycline. In addition to that, the mechanism of resistance is also multiple. So it's a very complex problem if one wants to develop molecular diagnostics, especially rapid diagnostics. So molecular point of care tests would be required for individualized treatments. They are being evaluated presently for the treatment with ciprofloxacin. They give a high sensitivity and specificity, and usually these are PCR tests. The technology is well established. And if one has a resistance determinant that for which only a few single nucleotide polymorphisms are required, then the accuracy of the test, in other words, the correlation with the test and phenotypic resistance, improves considerably. However, protocols will have to be developed in response to new AMR variants, and some of these tests are unsuitable for use on pharyngeal specimens, owing to cross-reaction with commensal Neisseria species. So what about ciprofloxacin? Basically, the levels of resistance vary around the world. This is recent WHO data for 2017, 2018. The red color indicates 70 to 100% resistance. The white means that there are no data available. And the pink areas are 30 to 69% resistance. Now that might seem terribly high, but in fact, it also means that anywhere from 40% and up of the isolates might in fact be susceptible to ciprofloxacin, in which case one could recycle this older antibiotic. Many, many studies have shown that genotype predicts resistance to ciprofloxacin. And the primary mutation predicting resistance to ciprofloxacin is this mutation in GRA S91F. There's almost 100% correlation with the presence of this mutation and resistance. The other interesting thing is that isolates with absolutely no mutations, you'll see this in the blue bar, in GRA are highly correlated with susceptibility. So looking at the MICs in comparison with mutations in these various genes that contribute to ciprofloxacin resistance, highly susceptible population, and this would be diagnostic. So this would be a wild type allele. This is a complex resistance allele, but this determinant S91F is predictive of MICs of 0.125 milligrams per liter and up. A variety, and this is only a smattering of non-commercial tests have been used to identify ciprofloxacin resistance and susceptibility in Neisseria gonorrhea. They're usually RT-PCR tests. They all target the SEER 991 locus and perhaps other loci. Some also are involved in identifying simultaneously Neisseria gonorrhea. All of these authors have used different primers, so there's a need for standardization. There's also a commercial test that has been FDA and CEIVD cleared. which basically detects Neisseria gonorrhea, as well as ciprofloxacin wild-type gene and the mutation at S91. This test has been used, for example, to look at gnat samples and has been shown to have a high sensitivity and specificity, over 98.6%. It has 100% sensitivity and specificity for detecting the wild-type gene as well as the mutation. And it has a very high sensitivity and specificity in detecting phenotypic resistance. So this knowledge has been used to basically develop a resistance-guided treatment. And this was done by Jeff Klausner's group, who used a PCR test to ascertain the mutant status of S91. And the results are very encouraging. So basically, the test was 100% sensitive and specific for all sites in predicting susceptibility to ciprofloxacin, followed by treatment with ciprofloxacin. What is the status of resistance with azithromycin? There are fewer RT-PCR assays developed for resistance. Basically, mutations in 2 genes are critical for ascertaining the development of resistance to ciprofloxacin. These are 23S, 2 SNPs, 23S ribosomal RNA. But it's complicated by the fact that other genes are also implicated in ciprofloxacin resistance. And so, although a commercial test is in development, when large populations of strains are being analyzed, these other genes could play a significant role in ascertaining the accuracy of the test. However, this is under development. It's also important to detect decreased susceptibility to ceftriaxone. Most of the world has sporadic isolations at the present time of ceftriaxone resistant isolates. Basically, Southeast Asia has the largest percentage of isolates, countries reporting decreased susceptibility to ceftriaxone. This is a very, very complex matter in actually trying to predict resistance through RT-PCR tests. There are multiple genes involved in ascertaining resistance, multiple mutations in those genes, and the genes act in concert, actually, to define levels of resistance. So people have taken various SNPs, combinations of genes, combinations of SNPs. In Pen-A, for example, sometimes one can have up to 80 SNPs, which are present in the gene and define resistance. And it's been reasonably accurate, but it's highly dependent on the population that's going to be tested. So what are the evolving methods for identifying mutations and predicting gonococcal AMR? Well, whole genome sequencing isn't evolving for surveillance, as Irene just pointed out. However, the use of MinION nanopore sequencing directly from urine samples and trying to predict resistance at the bench based on genome sequencing is still challenging. Bioinformatic tools have been developed to predict AMR. There's also people working on a multivariate linear regression analysis of WGS data sets to predict MIC, and that's been fairly accurate as a prediction, but is not yet available in terms of a point of care. Machine learning methods have been applied, different machine learning methods to try and identify emerging mutations and resistance. There have been molecular algorithms developed to try and predict resistance, especially to ceftriaxone, based on the presence of certain mutations in multiple genes in various populations. And finally, there are new point-of-care test methodologies that are in fact very promising and might be applied in the future to AMR detection as well, and which fulfill all the criteria of the WHO for rapid point-of-care testing. So in conclusion, regional variations in AMR may lead to POC tests to ciprofloxacin susceptibility and resistance. Is it possible that penicillin tests could be used? Irene didn't show, but in Canada, in the last surveillance, only 7% of the isolates tested overall were resistant to penicillin. In Saskatchewan, in some years, we have under 5% of the isolates tested resistant to penicillin. Could that be recycled? Molecular tests based on mutations in JARA are predictive of resistance to ciprofloxacin and have been used for individualized resistance-guided therapy. A commercial test is available in some countries. A commercial test for azithromycin resistance prediction is under development. Molecular tests and other strategies for AMR prediction are under active development, and genomics has revolutionized surveillance. And new molecular POC test formats for NG identification may be further developed for detecting gonococcal AMR. Thank you very much for your attention.
Thank you, Joanne. Very interesting to see how all things may come back. Learning a lot from these bugs, certainly posing new challenges. So now is the third part of the webinar, and we're going to be discussing maybe a vaccine. And it's my pleasure to introduce Dr. Ellen Marshall. She's the medical director at Virtue and a prof vaccinology at University of Adelaide School of Medicine. Professor Marshall is a medical researcher with specialist training in child health, public health, and vaccinology. In recognition of her research leadership, she was awarded the South Australia Science Award for Excellence in Research Collaboration in 2019, Research for the Public Good in 2010, and the National Public Health Association of Australia Fellowship in 2013. Her research was included in the NHMRC 10 of the Best Research Project for 2016. She was the recipient of the Australia Day Council Inspiring South Australian Women Award in 2020. Professor Marshall's research program is directed to address urgent priorities in infectious disease prevention in children and includes implementation research and clinical trials and investigational and licensed vaccine, infectious and social epidemiology, and public health. Our main interest includes meningococcal, human papillomavirus, influenza, and pertussis infections, and their prevention by immunization. So before I leave her the microphone, just to remind you that there are Q&A you can ask by pressing on the box Q&A, and we'll take time at the end to answer them. So now it's a real pleasure for me to let the microphone to Professor Marshall.
Okay, hopefully you can see that. Welcome everyone. I'm talking to you from Adelaide in South Australia around midnight. So what I wanted to do was obviously focus on the potential for a vaccine to protect against gonorrhea, but I'm also going to go through the impact of 4C-MenB on meningococcal disease, because that's what the primary indication of the vaccine is. So we'll just go through that and have a look at the impact of 4C-MenB on meningococcal disease and then on gonorrhea. And before we start, I'd just like to pay my respects to the Kaurna people, our First Nations people in Australia, who are the custodians of the land on which I'm speaking to you from, and acknowledge the deep feelings of attachment and relationship of the Kaurna people to country, and respect and value their past, present and ongoing connection to the land and cultural beliefs. Just to start with, I wanted to give you a little bit of background about our meningococcal immunisation program in Australia. We have a national immunisation program which provides the meningococcal ACWY vaccine at 12 months of age, and then for adolescents in year 10 at school, which is about 14 to 16 years of age. And 4C-MenB was provided for the first time in a national program from July 2020, only for Aboriginal and Torres Strait Islander, so again, our First Nations people, for infants in a 2+1 schedule, and a catch-up program for toddlers up to 2. The reason being they have 6 times an increased risk of meningococcal disease compared to non-Indigenous children in Australia. And then in South Australia, in addition to that, we have a state-funded 4C-MenB vaccine program for infants and adolescents. So in South Australia, we have a meningococcal vaccine program that covers all 5 common causes of meningococcal disease for both infants and adolescents. So that gives us a great opportunity to evaluate that program. The reason why we introduced a program in South Australia was, as a state, and you'll see a little diagram there, you'll see that South Australia is in pink there. We had the highest notification rate of meningococcal disease in Australia, and had so over the last 5 years. And WHO SAGE actually recommends that if you're getting to sort of 2 to 10 per 100,000 endemic disease, you should start thinking about whether you should introduce a meningococcal vaccine program. And while W and Y had increased around the world in 2017 and 18, in South Australia, we really had predominance of serogroup B, so that even throughout all the preceding years, B was the cause of over 80% of our cases, compared to about 50% nationally. The 2+1 schedule was under review by our Therapeutic Goods Administration. We already had a 3+1 schedule that had been approved, but obviously a 2+1, like they have in the UK, was going to be more cost effective. And isolates causing disease in Australia had been sent for MATS testing, so meningococcal antigen testing, to look at coverage, predicted coverage of 4C-MenB on disease in Australia. And you'll see in this little diagram, perhaps if I show the top one, where we actually had an increase in cases in adolescents over that period of time. I'm not sure why, but in South Australia, we have the same number of cases in adolescents as in infants. So adolescents was an important group. But if I show you then the MATS testing, you'll see the states are down on the x-axis there, and the predicted coverage you see for South Australia in the middle there. Our predicted coverage was 90%. So the vaccine should have protected against 90% of the isolates causing disease in South Australia. We had undertaken a large study to look at whether there was a herd immune impact, or was there a reduction in carriage that we would see with use of 4C-MenB. And after vaccinating 35,000 students in a randomised controlled trial, where schools were randomised to vaccination schools or non-vaccination schools, we found no impact on oropharyngeal carriage of disease-associated Neisseria meningitidis. So that's capsular strains that cause disease, no impact. So it was really important that our program set up for direct protection. You'll see there that in that graph, the way we decided what age groups to include in a free program in our state was to include any age group with an age-specific notification rate higher than average, and that average is represented by the red line, and that was average over the 2000 to 2017 period. And then any age represented by the green line that was above the red line, the average was included in the program. So you'll see in the table above the graph, there's two lines in purple. That's our continuing program in infants and 15, 16-year-olds. And then the catch-up programs are indicated there that have now been ceased, just with those two age groups ongoing. So we aim to estimate coverage, the vaccine impact and effectiveness against meningococcal disease, and then in infants, and then in adolescents, meningococcal disease and gonorrhea. We used published methodology to measure vaccine impact and effectiveness. With vaccine impact, we compared the incidence rate ratio of meningococcal disease in the pre and post vaccination period, very similar to how they've measured it, vaccine impact in the UK. And then vaccine effectiveness by the screening method and the case control method. And we used two different case control analyses. One is where we used controls from our Australian Immunisation register and used 20 controls to a case. And then for the gonorrhea case control analysis, we used chlamydia cases as controls. So the infant and child catch-up program, looking at coverage, impact and effectiveness. This is at 2 years post introduction of the program, which commenced in October 2018. You'll see that coverage was very high, 95% of one dose. So the dotted line here is one dose for each age group. The solid line is 2 doses and the dashed line is 3. And you'll see high coverage for 1 and 2 dose schedule, the priming schedule, with increase in the 3rd dose coverage over time. But as toddlers got older, you see coverage reduced, and that's really because just more of an opportunity around whether they're actually attending their GP at that age. They're less likely to compared to the under ones who are obviously attending for other immunisations. But I think it's important when we're looking at vaccine impact to see that coverage was lower in those older age groups. So looking at impact, which is really looking at reduction in disease in the pre and the post-vaccination period compared to pre-vaccination period, you'll see that for that the under ones, there was a significant reduction in the incidence rate ratio in the under ones, but not, although it was lower in the 1 and 2 year olds, it wasn't significant. And again, I think this is because of reduced coverage uptake, but also we had very few cases in that age group. But we did see a significant 60% reduction in the incidence of MenB vaccine in that under 1 year age group. When we looked at vaccine effectiveness, using both the screening method and the case control method, for infants, you'll see we have very high vaccine effectiveness of 94% using either methodology. We had two young children who had been vaccinated and developed meningococcal disease. The first one here had received 2 doses, but they had a risk factor for higher risk of meningococcal disease, and the second one had only had 1 dose. They both had isolates that would have been covered by the vaccine though. So in those two cases, they haven't really had an optimal number of doses. So adolescence, again, coverage and impact and effectiveness. And you'll see here, as predicted, that coverage is not as high in the adolescent age group. but it's quite good. It's about 77% there for 16-year-olds for 1 dose and close to 70% for 2 doses. But again, as adolescents get older, there's less likelihood that they will be vaccinated because they are attending their local GP rather than through the school program, where we have very high success with immunisation, the school immunisation program. So again, looking at vaccine impact, you'll see that we did see a reduction in the 15 to 18 years of age where we had high coverage, which was a 73% reduction in disease, in meningococcal B disease in that age group. And you'll see that it's, you know, like a statistician won't like me saying this, but borderline significant. The reason why that is you'll see our cases were quite low. The reason being that we had undertaken the Men-B carriage study and vaccinated 35,000 senior school students in that study. And you'll see we did see a significant reduction in that study, which is published in CID. So our numbers were quite low then when we introduced the state-funded program, and that's why we haven't seen a significant impact. But the 73%, 71% are very closely aligned, and that just shows you there that reduction that was significant in our B part of its study, looking at impact of forcing Men-B on carriage. The vaccine effectiveness in adolescents, we didn't have any cases in any vaccinated young people. And so vaccine effectiveness is around 100%, but we can't measure variance or confidence intervals because of the lack of vaccinated cases at 2 years. This is a bit really where, which is probably most relevant to this audience, looking at vaccine impact and effectiveness against gonorrhea with 4C-Men-B. And just really to make a disclaimer that the vaccine is indicated for active immunisation of individuals to protect against invasive meningococcal disease caused by Group B. It's not currently indicated in Australia or anywhere else to prevent gonococcal infection, but there's really mounting evidence that this seems to be another disease that the vaccine is able to provide some protection against. And that first data really came from New Zealand in around about 2004, 2000 to 2006. A meningococcal B vaccine was introduced called Men's B in New Zealand, and this was because there had been an epidemic of a specific meningococcal B strain, a clonal complex 4144. So the vaccine was designed specifically for that strain. The 4C-Men-B vaccine, of course, is different. It has the outer membrane vesicle from Men's B, but it also has outer membrane proteins that are found across a high proportion of serogroup B, but also other serogroups. So covers more strains that are circulating, causing disease. But with the Men's B vaccine, the specific strain-specific vaccine, because both gonorrhea and meningococcus, of course, are from the Neisseria family, Steve Black, who's I consider one of the grandfathers of vaccinology, suggested that they look at whether there had been any impact on gonorrhea over time in this adolescent age group who'd been vaccinated. And this is from a paper published by Helen Petrousis-Harris in New Zealand, showing that they did see an effect, a reduction in gonorrhea post introduction of that program. Now, we do see a reduction in gonorrhea compared to chlamydia, where it just continues to increase over that period, but it does seem to be a short-lived or perhaps 4 or 5 years that we're seeing this reduction, and then we see gonorrhea increasing again. And I'm not going to go through all of these results in this table, except just to point out that they found a vaccine effectiveness of 31% that was significant. And also, if you just look down the bottom, if you actually look at co-infection, so where there's both gonorrhea and chlamydia in cases compared to chlamydia, you'll see that the vaccine effectiveness is lower at 14%, which is just relevant for some of the other results I'm going to show you in a moment. So the next study was done in Canada, in Quebec. This was when there was an epidemic of, again, serogroup B, where 4-CMNB vaccine was introduced this time. So an opportunity to measure the impact of 4-CMNB in this young population. I think it was all children up to 20 years of age and adolescents up to 20 years of age were included in this program. And they measured vaccine impact, which is looking at the reduction in disease in your whole population and in your whole cohort, I should say. And you'll see here that there was a 59% reduction in gonorrhea in the 14 to 20 age group, which is the age group that had quite high coverage using 4-CMNB compared to the over 20-year age group, which were unvaccinated. You'll see that there are wide confidence intervals here, and it was a non-significant result. But again, it's consistent with the New Zealand findings around the men's B vaccine. The next study is a retrospective case control study, which was done in the US, New York City and Philadelphia. This is published at the moment just as an abstract from a presentation. There's not a lot of details about it, but they did estimate a vaccine effectiveness of 36%, using a very similar methodology to New Zealand with the case control approach, chlamydia as controls, and you'll see a very large number of cases of gonorrhea and chlamydia. And they then were able to link those to immunisation history on the immunisation register. Quite a small number of young people on their register had been vaccinated, but they found this significant vaccine effectiveness, again, around 36%. So very consistent with the New Zealand result. So I mentioned that we had undertaken a study to look at the effect of 4-CMNB on carriage of Neisseria meningitis in the throat. And although we didn't find a impact on disease-associated capsular meningococci, we found a significant reduction in unencapsulated meningococci. And so we've been kind of thinking about this and whether it's a way of understanding this impact on gonorrhea, or partly understanding this impact on gonorrhea, that the antibodies that are generated, we do know are cross-protective from work done by others, but also the fact that perhaps because the meningococcus is a mucosal bacteria, it likes to sit in that pharyngeal niche, and the gonococcus obviously is a mucosal infection. If there is no capsule, the outer membrane proteins are more likely to be exposed to antibodies, and perhaps that's partly why we're seeing a reduction in gonorrhea in those young people who are vaccinated. Additionally, the NHBA antigen is one of the antigens, one of the 3 antigens or proteins in the 4-CMNB, and NHBA antigen is exposed and quite well conserved in Neisseria gonorrhea. And again, so we would expect perhaps that we might see even improved reduction in gonorrhea with 4-CMNB than men's B, having these outer membrane proteins, and one that's specifically found in gonorrhea. And many of you will be aware that there's this high genetic homology between both Neisseria meningitidis, and Neisseria gonorrhea. So with all of that, we, of course, were interested in understanding any impact that we might see in our state program in South Australia. So we measured the impact of 4-CMB on gonorrhea. And you'll see here in this age group, 15 to 17 years, where the coverage was high compared to the older age groups where coverage was quite low. You'll see we did see a reduction in the post-vaccination period compared to the pre-vaccination period, with a 24% reduction in gonorrhea in that younger age group. However, again, our findings, this is not a significant difference, and the confidence intervals include 1, based really on our fairly low numbers of gonorrhea cases in that 15 to 17 year age group. In South Australia, we don't see many cases in that age group. Most of them are, the highest rates are in the 20 to 30 year age group. So we then looked at did a case control analysis to look at vaccine effectiveness, and just to acknowledge Dr. Bing Wang, who's one of my postdocs who did the analysis. Again, we followed the New Zealand methodology quite closely, using chlamydia as controls and undertaking a logistic regression. And our vaccine effectiveness of, was 32.6 against gonorrhea for greater than or equal to 1 dose, and pretty much the same for exactly 2 doses. But we did have quite high coverage of 2 doses, which is why I think these 2 results are very similar. And then we also undertook a case control analysis, as I'd mentioned before, using controls from the Australian Immunisation Register. Now this gave us a higher VE, and as did the screening method. But we really consider both these methodologies subject to bias and confounding, that because we don't really have enough variables to be able to adjust adequately. So we really believe the primary analysis using chlamydia controls is more robust, and again is more consistent with the other studies. So we're interested in this question about co-infection, whether we would see a reduction in vaccine effectiveness in co-infection cases, and you'll see here we actually didn't see that. Where we looked at gonorrhea and co-infection, we really believe this is a better kind of real world assessment, because you're always going to have people who are co-infected, rather than separate them out. But if you do separate out and remove all your co-infection cases, you have a VE of 35%. Now, obviously, all these are very similar in confidence intervals overlap, but perhaps cases of co-infection do reduce the vaccine effectiveness a little bit. So we've gone on to undertake a study in the Northern Territory, which is on this map, the northern part of Australia there, as indicated. And in the Northern Territory, rates of gonorrhea are much higher than anywhere else in Australia. And not only that, the highest rates are in the 14 to 19-year-olds. In some communities, some remote communities, the incidence is as high as, the notification rate is as high as 17,000 per 100,000 in the 15 to 19-year age group. And where you're giving the 4-CMB vaccine to protect against meningococcal disease in that age group, it's really the perfect place to be measuring the impact of gonorrhea in this age group as well. They also have high rates of, quite high rates of disseminated gonococcal infection. And again, the highest rates of disseminated infection are in that 15 to 19-year age group. So we're looking at the impact again on meningococcal B disease and gonorrhea in the 14 to 19-year-olds. We've recruited 1,300 adolescents to date, and we're hoping to recruit 5,000. And in addition to that, there's quite low genetic diversity of the gonococcus in remote areas in the Northern Territory, and some of the Pore expressed is very close homologous to the meningococcal strains causing disease in the Northern Territory as well. I'm not going to go into any more detail about that study unless people are interested, but you can find out as much as you like from the website there, www.bepartofitnt.com.au. So just to summarise then, we have in South Australia a program that covers meningococcal disease prevention across all the strains that cause disease in infants and adolescents, based on providing direct protection against meningococcal disease. We've shown high vaccine effectiveness against meningococcal B disease and reduction in disease in both age groups. And I think from the evidence I've shown you tonight, there is really this accumulating evidence of vaccine effectiveness against gonorrhea of about 30 to 40%. And we'll continue to undertake another year's evaluation of our state-based program against gonorrhea as cases accumulate in that younger age group, and evaluation of our Northern Territory study, and then we'll combine the South Australian and the Northern Territory data so that we're well powered to look at this. So just to thank all our partners, and based on our findings of the evaluation of the program in South Australia, because we had such high effectiveness, our program will continue now indefinitely. And that's it. Thank you.
Thank you, Professor Marshall, for your nice presentation and giving us hope in terms of prevention of gonorrhea. Question that is asked, is there a point where the cost benefit ratio would support, even if it's not at the same level of?
Yeah. Sorry, I missed you there, Mark. I think my screen froze, but you were asking about the cost effectiveness?
Yes. Is there a level that would permit the cost to be taken by a national program? And we know this is not the kind of efficacy that we see with HPV vaccine, but still it looks very enticing to invest in such a prevention program.
Yeah, we are doing a cost effectiveness analysis of the state program at the moment, and we've got including the costs of meningococcal disease and gonorrhea. Gonorrhea is not it's common, but it's not particularly expensive to treat, except for the problems you get, of course, with antibiotic resistance and also with disseminated infection. That becomes a much more expensive cost. But I do think that in high-risk groups particularly, I expect it will be cost-effective. So I don't know at this stage whether or not we'll come out with a cost-effectiveness for all young people in Australia, but particularly for our high-risk young Aboriginal people, I expect it will be cost-effective. The big cost is really around the management of meningococcal disease and the sequelae from meningococcal disease, but the gonorrhea is going to add, an important component to it as well.
For those people that want to ask questions, there's the Q&A box that you need to press before entering your question. There's a question about the high rate of disseminated disease. Maybe Irene can answer. Are we seeing the same rate? It seems that you have much more cases that we see in Canada.
Yeah.
Irene, would you have an answer?
Yeah. Thanks, Mark. We actually are seeing more and more cases of disseminated gonorrhea, which is quite alarming. But we don't have the data, the actual numbers, like Dr. Marshall was able to present. So that was a surprising number to see.
Yeah, it is, especially that young age group. There is a really nice paper published showing that data for the top end of the Northern Territory. There's a second paper on the Central Australia, but at the top end has a lot of information, a lot of detail about the disseminated disease.
There's a question about treatment. Someone says the United States has moved to 500 milligram IM ceftriaxone monotherapy. Is PAC moving that way too? So maybe Irene or Joanne?
Well, I can give you a little bit of information. I know that the STI treatment guideline group for Canada is reviewing the chapter, and so that is ongoing. Unfortunately, of course, the pandemic slowed down the progress on that, but they are revisiting the treatment options for Canada as well.
A question about the changes we have seen in some provinces in Canada, moving for the treatment of chlamydia from azithromycin to doxycycline. Can you predict from our data that this will improve the situation for gonorrhea?
It's possible that when you know that pressure is lessened and less azithromycin is being used for chlamydia, it's possible that we'll see a reduction. We'll have to wait and see, keep our surveillance ongoing, and we'll see what happens.
Someone is asking about Australia moving forward. Do you know if the data at this moment is sufficient for an official program, or there's still more information to come before Australia moves to a national program? Yeah,
there's some really nice studies that are about to start or ongoing, and I think I think we'll get to the stage that we'll have a lot of really robust evidence around this. I think we're sort of in the middle of collecting that at the moment. I think the Northern Territory study will be really important because it's in a very high-risk group and it's the highest risk are in the same age group as you're giving the meningococcal B vaccine to anyway. There's another study, a randomized control trial looking at the impact of 4-CMV on very high-risk young adults. So these are young adults who have recurrent episodes of gonorrhea, but because of COVID, that hasn't even been able to get started yet. So, that's going to take a couple of years, I think. And then I think there is another study in the States. So I think it's, as we just generate more evidence and consistency in the evidence, I think it will be difficult to ignore when we don't have any other vaccines for gonorrhea yet. And because we're getting a continuing problem with antibiotic resistance, I think there'll be quite a strong case, but it's not something being looked at the moment in Australia.
And because of the relative success that we see with the MenB vaccine to prevent gonorrhea, might it be something dangerous so that company would stop developing new treatment for gonorrhea?
No, I don't think so. I think, vaccines, gonorrhea specific vaccines have been difficult to generate for a number of reasons, but there is quite a lot of work going into that at the moment, identifying which antigens are going to be important for a robust immune response. So I think because the 4-CMV is 30 to 40% effective, it's certainly not going to get rid of all disease. So we still need, antibiotics. We're still going to need to develop gonococcal vaccines that are specific.
Thank you. I think it's all we had, all the time we had. So I would like to thank Professor Marshall for her great presentation, Irene and Joanne for introducing Canadian picture in the webinar. And in finishing, I would like to thank GlaxoSmithKline, who provided an unconditional support for this webinar. This webinar was taped, and if you are interested to share with colleagues or to look at the webinar again, it will be available on IUSTI Canada website. I would like also to thank Canadian Public Health Association, Greg Benny and Laura Bouchard, who provided technical support for this webinar. So thank you all for being there, and we'll be in touch soon for our next webinar for IUSTI Canada.
Thanks for the invitation. Bye.
My pleasure. Thanks, everyone.